But that's a very, very low-level representation. That's a representation of the world, kind of sort of in the same way as this is a representation of a cat. It's just a picture of a cat. I mean, if you took this and distorted it a bit. Okay? So, but certainly not in the way that this is a representation of a cat, and certainly not in the way that this or this is a representation of a cat. So this is a type of neural representation. And it's literally a map. So these low-level sensory areas literally contain maps of the world. In this case, a visual map of the world. In your auditory brain, you've got an auditory map of the world, sort of. You've got a map of your body and your somatosensory cortex. You've probably seen these pictures of like the little person with the big hands and lips who's kind of stretched along your brain. I'll show a picture of that in a different class if you haven't seen it. So you literally have these maps, but this is, this is the simplest, lowest level, most literal-minded type of representation. But even this actually obscures a lot. I mean, there's a lot of stuff going on in primary visual cortex that you're not seeing in this picture. But in amongst all that other stuff, there's a map. So if this was what mental representations, what neural representations were like, then decoding the brain or understanding neural activation would be kind of easy. But if you did a similar kind of thing, if a similar kind of thing worked for higher level representations, suppose representation of a number worked like this, then you'd get brain images that look kind of like this, and we don't get that. So this is a Photoshop job. So there's not by me, by some others. So this is actually the logo for SPM2, if any of you. We're going to be exploring using SPM8, so this is kind of an old version. So, and you can see there's literally, there's this kind of low-level representation. It's not just that they don't exist for high-level representations, or they don't exist that we're aware of, but they kind of couldn't even exist. How could you possibly have, a picture of the number 2 or a picture of the concept 2 in your brain? It's not the kind of thing you can really do a picture of, right? How could you possibly have something like this kind of just, literally represented in your brain? So once you get past a certain very basic level of maps, the question of what is a representation becomes a lot more difficult. But it doesn't mean that it's intractable, and in fact, it's possible to access information about these higher level representations as well, but it just isn't going to come out in anything like this kind of pictographic sort of form. But I think it's quite striking that in certain low-level representations it actually does. And apparently, the story is that when this image was first shown at a Society for Neuroscience conference, I guess in 1981, there was a standing ovation from the floor. That's what I heard. I don't know if that's true. Yeah.
I think you might be being a little bit unfair, because the information that is relevant about this thing that we're trying to find a representation of is just spatial information, right? So there's just there's some set of spatial relationships that in the brain recapitulate spatial representations out in the world, right? So the fact that it seems that the idea of thinking that the representation of the number 2 has to be like a picture of a 2 glyph in the brain, right? I mean, 2 is many more things than just
that's obviously
right. But it could very well be the case that you have a nice linear ordering of numerical magnitude, or
it could be, and in fact, and I don't, maybe you're thinking of this. There was actually a paper that came out in Science a few months ago that actually claimed to find such a map.
I didn't know that.
Oh, okay. Which we should, which would be an interesting thing to talk about. And then there's some other papers some papers that came out saying, oh no, those guys did it all wrong, which happens a lot, by the way. Okay. So it also, and this is, I think, especially true for undergraduates, but for all of you, as we read stuff in the course, do not be alarmed if, because it will happen, you read one person who says, well, we all know that X, and then someone else says, we all know that not X, and that this is kind of, research. This is where people are very uncertain about what's going on, and there's not agreement in the field, so you're going to find that kind of thing. Anyway, that would actually be an interesting thing to look at too. Well, I agree with you. So there could be map-like representations but kind of more abstract maps, maybe. And,
I mean,
or...
this is an abstract map. It just so happens that, you know, we're appreciating the structure in the same way.
Right. No. Okay. That's right. I mean, this, for instance, has a high level of distortion, for instance. So it's not. It's a straight picture. Well, this is actually a very interesting open question. So nobody knows how far this idea of maps go. But I think it's worth pointing out that it may not necessarily be a spatial thing. So for instance, this is kind of going a little bit off the topic of the kind of things that you can look at with fMRI. But there's a big debate in neuroscience about what you can represent with what's called a rate code, which is like the rate at which neurons spike per second, versus temporal code, which is, you know, the information is really kept is really stored somehow in the way that different neurons kind of spike in synchrony with each other. And that's a very, very live and open debate, and there's really no agreement about that. So there probably is some kind of information in this kind of synchrony code, probably, maybe. And so that's a very different format of representation. That's not spatial at all, that's like a temporal thing. And of course in speech, there's a lot of stuff represented temporally as well. So you know, once you get to something being represented in the time domain, then it's kind of even less a map. So these are open questions. But I think we can agree that, and I'm not requiring, because, you know, as I say, these are open questions, so there's no reason for us to all agree. But let's just say that at lower levels, there's things that really do look like maps, and at higher levels, there may or may not be, but it's certainly a lot less clear. So here's one way of thinking about mental representations, which is just it kind of totally bypasses this question of how is it actually represented, and just says, okay, I'm just going to say that there's somehow information about the world that's in our heads. So if there's some shape or form in which information about the world is in our heads, I'm going to call that representation. So you might call that like a kind of operationalized version of representation. And that leads to all kinds of questions like, you know, how does the information get there? How does it lead to behavior? How is it processed? How is it structured? And we're going to look at some of those questions. So the kind of worrying side of this, if this kind of thing worries you, is the very question of how something is represented in the brain gets very unclear very quickly. But the more positive side is that you can actually, you know, ask questions about it even if you don't really necessarily know the answer. You can ask the question of, is there information in there which I can get out, independently, or at least without having to have finished answering the kind of what seems like the logically preceding question of, you know, how is it represented? And in some ways, it's kind of surprising that that you can try and pull out information without even necessarily knowing kind of, you know, how the information is written. So, okay, I promise this is the last bit of, for a little while, of kind of philosophical type stuff, but I do like this this picture. Have any of you encountered the idea of the Cartesian Theater? Any of you read any? You don't have to put your hand, but any of you read any Dennett? Daniel Dennett, he's like a very... So, okay. So if you ever want to read anyone in philosophy of the mind, if you ever find yourself lying awake at night pondering some of these questions, pretty much the best person you could read in mind, any other opinion, people's opinion, is Daniel Dennett, who's a very eminent philosopher who talks a lot about cognitive science. A lot of people actually kind of get interested in cognitive, I'm one of them, get interested in cognitive science from reading his stuff, actually. And he has a very, very nice, he uses what he calls intuition pumps, in which he tries to basically take your ideas that are kind of, you know, seem like they're well formed and say, well, have you thought about it like this? And so one of his best intuition pumps is what he calls the Cartesian Theater. And I think this is a useful way of thinking about, if you like, how not to think about mental representation. Okay, so we just saw, okay, that there is literally, in a certain sense, a picture of the world in your head. Okay, well, that's great. Now we figured out how the world is represented, right? Because if all you need is, this is why it's so unsatisfying, but it's maybe just the first step, to say, well, if there's information about the world in your head, then that's a representation. Okay? Well, here's information about the world in your head. This is the world, in this case, this kind of flashing checkerboardy thing. And here's literally a picture of it in the cat's head, and there's one just like this in your head. if you were to look at such a checkerboard. And so there you go, there's information that's gone from out there to in here. So now we have a representation. Well, what's the problem with that? Well, if you have the same picture again, who's looking at the picture, right? Now, it kind of sort of feels like we're this, we're kind of in our heads looking out, watching this kind of movie screen of the world, right? I mean, it does. But there's a sense in which logically it just cannot be right, okay? And that's what Dennett calls the Cartesian Theater, because, well, does anyone want to explain what is the problem with this picture? Maybe I shouldn't take you for granted. This is also not a trick question, by the way. Well, it might look like one. It's not. Why can't this be how our perception of the world works? Yeah, someone who hasn't. Yeah, hey, Albert.
Well, my first thought is that there would have to be another theater inside of the man in the theater and another theater.
Yep. Yeah, exactly.
That.
Exactly. So this is what gets called an infinite regress. So, if you say, oh well, we kind of figured it out. There's a picture of the world inside their heads, so now we got it. Well, you say, well, what looks at the picture? Okay, well, whatever looks at the picture, it can't just do this all over again, because then something's going to look at that picture, something's going to look at that picture. And so even though it might kind of sort of feel as if what it means to have representation is to have this kind of, little movie of the world going on inside your head that we're watching, that can't work because, or at least that in that form it can't work, because you'd have an infinite regress. So, and so the term that you'll see actually probably in, I don't know if you've seen any of the papers that we've read so far, maybe not, but you'll see it if you read enough kind of cognitive science, is this term homunculi, which is this Latin for little man. Okay? And the idea is that if you explain something by saying, well, in effect, I mean, no one ever explicitly says this, or not usually, but if you explain something by saying, yeah, well, the way this works is that the information gets in there and then it gets read out. Well, you say, well, who's reading it out? Well, if it's kind of implicit, well, some kind of clever little man who's sitting in your head. The clever little man often ends up in prefrontal cortex. This is a very, very common thing. If you ever see, for instance, it's a very unfortunate tendency in fMRI papers, less so than it used to be, to say, the neural basis of X is, you put people in the scanner and they do a crossword puzzle or something. And you say, okay, so the neural basis of solving a crossword puzzle is that, it makes prefrontal cortex light up. So prefrontal cortex is, the thing that does crossword puzzles, okay? But you haven't really increased our understanding of how does the brain manage to do crossword puzzles. You just said, well, there's some clever stuff here. There's a little man who sits here, or a little person sits here, okay, who does crossword puzzles. Okay, so this is so when you're thinking about mental representations, if this is too philosophical for you, then I'm sorry, but I personally think this is useful to think about. But there's no reason why you're obliged to agree with that. So if your explanation basically involves there being this kind of little clever thing that does the job, but we don't really know how it works, then you have basically a homuncular explanation, which is not really an explanation at all. And so then it actually does one other really nice thing, where he says, well, does this mean that any explanation that it looks kind of homuncular at all is definitely wrong and definitely circular and definitely has this infinite regress? And he says, well, no, not necessarily. Okay? So he says, well, what if the little homunculus is a little bit, suppose you have this whole chain of them, right? Alberto was saying you have this infinite chain. What if the little homunculus is, this one's a bit smaller and a bit stupider than the previous one? Okay? And then this one is made out of other little homunculi, but they're a little bit smaller and a little bit stupider than he is. And each time they get a bit smaller and a bit stupider, then eventually the homunculi could just be made out of quite small and quite stupid things like, say, individual neurons. Although individual neurons actually can do a lot. But so then you'd say, well, if you've actually managed to reduce your explanation down to things that are sufficiently simple, even if it's had to go through this kind of pathway of homuncular type thing, then you've discharged your homunculi. So the problem with an explanation is not really that it has homunculi in it, but it has homunculi which are undischarged, meaning that they haven't been broken down into sufficiently small and stupid stuff. Anyway, so that's just some ways of thinking about mental representation. I kind of get the feel, just looking around, I kind of feel like that last part kind of made a lot of you tune out, which is okay. This is you can think about fMRI without having to think about these kind of things. Okay, so let me just quickly recap some more concrete stuff. So that's one set of reasons why it's difficult to think about representations, and the other is because the real action is being done quite far away from what we're measuring, and we already talked about this a bit, but I'm just going to go over it again. So, well, actually, I don't think we talked about this. So, there's another idea of representation is this concept of a grandmother cell. Have any of you heard this concept of a grandmother cell before? Okay, so the idea is that, just as you could have brain areas specialized for, I mean, you probably don't, but suppose you had brain areas specialized for a particular thing, or within those brain areas there might be neurons specialized for particular aspects of that thing. And if you follow this logic far enough, you might say, well, eventually there might be a single neuron that's literally just specialized for recognizing your grandmother. Okay? And you might think, well, that's the most ridiculous idea in the world. Of course, that couldn't possibly be true. What if that one cell died, then you wouldn't be able to recognize your grandmother anymore. But then people might say, well, it's not just a neuron, but there might be a population of neurons. And there's actually evidence, believe it or not, that for instance, there's some neurons that do seem to be tuned to very, very, very specific things. There's a famous study recording from open human brains of epilepsy patients, actually, with neurons that responded specifically to Jennifer Aniston. Have any of you heard of that study? So this is also an open area. But the key point here is that the real information processing is being done by neurons, not by blood, not by voxels. So another reason why it's hard to get to representations. These are all reasons why it's hard, but I think, as we're going to see, despite all these obstacles, people can actually sort of get to some representations, I would argue. But it involves overcoming some of these challenges. So if you really, really, really wanted to understand how information is being represented in the brain, you'd somehow have to read the information from millions of individual neurons all at once. Have any of you heard of this brain initiative that Obama recently announced? Okay, so this is also a controversial idea. The idea is to develop technologies. There's I think $100 million being devoted to it so far, which sounds like a lot. In terms of neuroscience funding, it's actually not that much. To develop technologies for being able to record from lots and lots and lots of neurons at once. At the moment, people can record from a few dozen, maybe. So, and then the question is, if we had all that data, would we be able to understand it? That's an interesting, difficult question. But certainly, if we really wanted to get to where the action was, that would at least be a helpful step. And we're certainly not getting that with fMRI. We also discussed this a little bit. This is partly recap. The neurons are much faster. So you answered this question of what's the hemodynamic response function, and the key point of that is it takes several seconds. Okay, it takes about 5 or 6 seconds to go up. We're going to explore next week. We're going to explore a lot about hemodynamic response functions and what they look like and how they might overlap with each other and add up and things like this. And we're going to do that more in computer exercises. But the key point here is it is much, much slower than things that is going on in the brain. Neurons operate on the timescale of milliseconds. If our information processing operated on this timescale of seconds, we'd all be dead very quickly, right? Okay. So we can respond to things. I mean, it takes us maybe a couple of hundred milliseconds, but that's pretty fast. The other neurons are small. fMRI measures are big. So there's at least 100 billion neurons in the brain. Probably more depending on how you count it, depending what type of cell types you count. And a voxel, even if you have a good resolution, is at least several millimeters across. So there's literally millions and millions of neurons in there. And they might be representing different things. Okay, so here's, this sounds like negative, but I want to emphasize that despite all these obstacles, we actually can ask interesting questions. But I think it's very important to actually be aware that these are real obstacles. So different neurons may represent different things, and they're all together in a voxel. We're just bunching them all together. And then we also discussed this a little bit, but I'm just going to go into a little bit more detail about this. and this is really the essence of the Haxby paper, talking about distributed and overlapping representations. So I showed you this picture before. Quentin very helpfully pointed out that I had these two the wrong way around. Thank you very much for fixing that. So there are some representations in the brain that are sort of segregated, although probably not as segregated as they look. But most representations in the brain are all kind of sitting on top of each other, especially if they're representations of the same kind of thing. And so that's what the Haxby talked about, overlapping representations. Before this paper came along, it really was kind of difficult to look at overlapping representations because you had to look for activation that was either here or here. And if the activations were sitting on top of each other, well, I'll show you why that was a problem in just a minute. So here's a kind of cartoon of overlapping representations. This is not the kind that Haxby looked at. This is more the kind of I and other people have looked at. Okay, so suppose I've got different types of speech being represented in the brain. So I might have, say, the sound ra, but I might have a different speech sound, say la. Well, that's going to be activating more or less the same part of the brain, right? Because if some part of the brain is specialized for processing speech sounds, it would be incredibly wasteful to have different chunks of tissue for different speech sounds, right? They're all going to get processed by this kind of speech sound processing area. So all of these different representations are all going to be right on top of each other. You're not going to have a ra area over here and a la area over here. So all these different, in this case, phonemes are all going to be kind of sitting on top of each other in the brain. So you have this kind of representational pileup of all these overlapping representations, and how are you going to kind of untangle that pileup? So that's the challenge. And in the most standard, when you remember when we were just looking at individual voxels, say in the Haxby time course, the voxel goes up, the voxel goes down, the voxel goes up, the voxel goes down. So you're just looking at changes in activation intensity. But the problem is the different representations may produce the same overall level of activation intensity. So suppose, here's a little kind of cartoon of suppose this is some voxel, so like the brighter it is colored in, the like more intense voxels. So this is like a bunch of like say 9 voxels, and this is stimulus A. It might be you're hearing ra, okay? And in the cartoon, this is stimulus B, so you're hearing bar, and I've just jiggled around these squares, okay? So the total amount of activation intensity in this little chunk of brain, this might be just a single voxel, or this might be a bunch of voxels, okay? The total amount of activation intensity is exactly the same, but the pattern is different. So this is why I'm going on about patterns, that if you're able to look at the pattern being different, then you can see these differences that may not be apparent in activation intensity. And that was, the Haxby paper was kind of the first paper to do that. So the average activation is the same for both stimuli. So here's a kind of illustration of why this same average activation is a problem. So we saw that fMRI activation is very noisy. Okay? And generally speaking, you want to get rid of that noise. And a good way of getting rid of noise is just to kind of average it out. Right? So the idea is that if you keep on having the same thing over and over again, the noise is just going to kind of flap around differently every time, and eventually that will all cancel out. It will all average to 0, but the actual signal will still be there. So one way of averaging things out is basically to say, let's take the average activation across, say, some local neighborhood of voxels and just find the average of it, which is basically just saying, let's kind of apply a blurring to this. If you just kind of blur this enough, if you spatially smooth this enough, then you'll be kind of averaging them all together. Just like if you kind of, scrunch up your eyes, or take off your glasses if you have glasses, when you blur your vision, you're basically just kind of averaging together like neighboring areas. So the problem with that, as you can see, is, well, okay, that averaging can be quite good for getting rid of noise, but how do you know you're only getting rid of noise? You might be getting rid of signal. What even is signal? What even is noise in this case? It's not necessarily clear. So if I take these and I just calculate the local average activation, because I just jiggle the squares around a bit, they come to exactly the same thing. So I might say, oh, well, here's the average activation for stimulus A, here's the average activation for stimulus B. I can just kind of, subtract one from the other, and I say, oh, there's no difference whatsoever. So clearly the brain this part of the brain doesn't know anything about the difference between stimulus A and stimulus B. Well, in that case, in this particular instance, that's not right, because the brain is doing something different for stimulus A and stimulus B, but it's doing it differently in the pattern of activation, but not in the intensity of activation. So, so that's why, that's essentially, that's one of the, there's actually other reasons as well, but that's one of the reasons why looking at patterns really opens up new kinds of questions. But when we look at, we're going to go through kind of what are the standard steps where you take fMRI activation and you kind of massage it into a form that's easier to work with. And in fact, one of the standard steps, which you would actually skip if you're doing a pattern-based analysis, is to apply spatial smoothing, which is basically just to try and get rid of this, what seems like noise, but may actually not be noise. So, in fact, one of the reasons why, for at least 10 years of fMRI, people didn't really even think to look at these patterns, because one of the first things they did was to smooth them all away. And it seemed like a very reasonable thing, but turned out not to be. So it's entirely likely that there's things that we're doing in fMRI analysis right now that are missing things that are right under our noses, because that did happen in the field for a long time. So that means that, you know, there's potentially interesting stuff out there. We just don't know what it is yet. And so there's a whole bunch of different studies. We're going to look at some of these which have kind of done examples of this. I did a study like this, some of these other people did too. Okay, so now here's I think we're going to have to talk more about this in coming weeks, but here's another sense in which just looking at, going from looking at individual voxels to looking at patterns of voxels gives you something different. It's not just that you have this greater sensitivity because you're now able to see patterns that might have the same overall activation. That's one reason. But another is that now you can see all kinds of structure. And I personally find this a very kind of interesting direction to look at. So if you're just looking at individual voxel, it goes up, it goes down, it goes up, it goes down. And so that's the kind of jargon term for that is univariate, one variable. And massively univariate means you're looking at lots of single variables all at the same time. So you might have a whole brain full of voxels, 40,000 of them, and you're looking at all of them. Does it go up, does it go down, does it go up or down in sync with the signal? But each one is being looked at individually. That is actually the standard way of doing fMRI analysis, and that's how it's been done for a long time. And that's great for seeing whether each individual voxel is going up and down in sync with the stimulus. So that's the jargon term for that is univariate. So the multivariate means looking at multiple variables at the same time. In other words, looking across not just one voxel, but the pattern across multiple voxels. And that's one of the main things we're going to look at in this course. And so then something very different happens. That instead of the activation just going up and down, then things can be more similar to each other or dissimilar to each other. Remember in like a plot of basketball players and sumo wrestlers, you had kind of 2D space and things could be closer or further away from each other in the space. So that's a kind of similarity structure, and that's something that people look at a lot in fMRI. So so next week we're going to look more at, let me skip to, okay, so next week we're going to look in more detail using MATLAB at these things like hemodynamic response function, time course, design matrix, very simple classifier, and then after that we're going to start looking a lot more at things like neural similarity structure. And just to remind you, so, and I'll send out this in an email, but please send me your initial thoughts on, before like the first class of next week, I guess, your initial thoughts on class project, and they can be very preliminary. That's the idea, to start developing. Thanks very much.
Do you want a break, or do you want to?
Something like 5 minutes.
Okay, so just continue.
I was thinking all the time when we were talking about the health study was the long-standing debate between cultural evolution.
Now that's one of the very few things that actually did get resolved, kind of. So.
Well, it sort of just stopped, I think, after like 35 years. But if you ask, I mean, I think Volition never No, never ever. And I think what Carlson did was he showed, like, for 30 years that we can collect more and more data that are in support of this sort of, as Volition called it, picture theory. But it made me think of, I don't know if you remember, he had a 99 cent puzzle where they showed that mentally imagine like that cat. mentally imagine a cat, and you look at the responses in view one, they look similar to what it would look like sort of in a rhythmotopic manner.
Right,
And
right.
as you zoom in on your mental image, it becomes more and more
yeah, I remember that
parafoveal.
one. Yeah,
Then
then they would get people to do like a telephone, so they'll get people to do like very detailed things like
Yeah.
which side of the receiver is higher up, is it the left side
Yep.
or the right side?
Yeah. And you know, they would show that you get these sort of response properties that you get with using actual stimuli.
Yeah.
Actual visual stimuli.
Yeah, no, that's very interesting, and now that's even been, you know, better and better established, so he was definitely on the